Every living thing has DNA, including plants, animals, bacteria, and us, humans. Let's begin our journey by zooming into this remarkable molecule, composed of a double helix structure which carries the genetic instructions that make each living organism unique. At its core, DNA is made up of 4 nucleotides: adenine, thymine, cytosine, and guanine, paired together to form the rungs of its twisted ladder. Adenine will always bind with thymine, and cytosine will always bind to guanine. But DNA is more than just a sequence of these bases. It holds complex layers of information. Throughout this webinar, we'll explore how scientists have figured out how to determine the order of DNA nucleotides in a process called sequencing. So, how are we analyzing DNA? To understand this, we will take you on a journey through the decades, and the first stop is 1977. Starting with the Sanger sequencing method. This was first developed by Frederick Sanger in 1977. This technique uses radio-labeled special nucleotides that are altered by missing a functional group required for elongating DNA. Here is how it works. These modified nucleotides are added to a mix containing template DNA, a short single strand of DNA to be used as a primer, and single DNA nucleotides. The reaction starts by separating the 2 strands of the DNA template at a high temperature and lowering it to allow the primer to bind. The primer serves as a starting point for DNA synthesis. Once activated, DNA polymerase is added and it starts to elongate the primer by adding nucleotides one by one, until it adds the modified nucleotide that stops the elongation at different lengths. This reaction is performed in parallel mixes for the 4 DNA nucleotides. It is then run on a gel to see the signals emitted by the radio-labeled nucleotides, allowing us to read the DNA sequence from the original template. This pioneering technique revolutionized the field of molecular biology and became the most widely used method for DNA sequencing. Over the years, it has undergone numerous improvements and has remained a gold standard for validating results and sequencing smaller genomic regions with high precision. The second generation of sequencing was brought on by Pyrosequencing in 1996. This technique differs from Sanger sequencing because instead of halting the elongation of the DNA strand, it relies on detecting the light as a signal when a new nucleotide is incorporated. It does this with the detection of a pyrophosphate molecule. Here is how it works. First, it starts with DNA that is copied multiple times. The DNA is combined with many things including a primer, several enzymes, and substrates. One type of nucleotide is added at a time, an A, C, G, or T. If the added nucleotide is complementary to the base in the DNA strand, DNA polymerase will incorporate it, releasing a pyrophosphate. This pyrophosphate triggers a reaction that creates energy, which powers an enzyme called luciferase. The luciferase enzyme produces a flash of light that is detected by a detector, and the sequence of flashes reveals the order of the nucleotides in the DNA strand. While both pyrosequencing and Sanger sequencing accomplish the same thing, this method was able to do it in a less complex, faster way. And a huge bonus, this technique can process different DNA strands in parallel and can be automated. Imagine how much time a scientist could save. During the 1990s, scientists needed to sequence large genomes more reliably to be able to sequence the human genome. For this, they came up with the hierarchical shotgun sequencing, a variation of the whole genome shotgun sequencing method. Here is how it works. First, it starts by breaking DNA into small pieces that are inserted into bacterial artificial chromosome vectors. These amplify the DNA sequences into multiple clones to isolate them afterward. Next, restriction enzymes are used to recognize specific DNA sequences and cut them at different lengths. Each of these fragments are compared to identify overlapping sequences and determine the order of each clone. After this, sequencing is performed based on their overlapping sequences. This was the strategy used to determine the original human reference genome in the Human Genome Project, an international collaboration to produce and make a freely available human genome sequence. This project started in 1990, with the first draft published in 2001, and it was officially completed in 2003. The big leap in technology came from Illumina through their launch of their genome analyzer in 2006. This technology, called short-read sequencing, was the first to sequence millions of short DNA fragments in parallel. Here is how it works. DNA is broken up into small fragments of 50 to 300 nucleotide pairs. Next, we add small artificial DNA sequences, called adapters, to the end of our fragments. The DNA strands are then denatured to get single strands. The adapter has sequences that act like glue to attach to a surface called the flow cell. Now that our DNA is anchored in place, the fragments are copied multiple times through a process called bridge amplification, creating clusters of identical DNA copies. These copies are what create a strong signal. In this process, special nucleotides are used that are fluorescently labeled so that each nucleotide has an identifying color. As the sequencing progresses, the nucleotides emit color captured by a detector. To this day, sequencing of short reads remain the most widely used form of next-generation sequencing due to its high capacity to analyze large amounts of DNA and its accuracy. In 2007, chromatin immunoprecipitation sequencing, also called ChIP-seq technique, was developed to determine where proteins bind across the DNA sequence to help us better understand gene regulation. Here is how it works. The classic ChIP-seq starts by cross-linking proteins to DNA using UV light. The DNA is then fragmented, and these DNA protein complexes are precipitated with special molecules called antibodies that specifically bind to the protein of interest. After immunoprecipitation, the next step is to reverse the cross-links by using high salt concentration and heat. Now, DNA can be purified, sequenced, and mapped to a genome, revealing the DNA sequence regions bound to our protein of interest. This method gives us a picture of where proteins, such as transcription factors, bind to DNA. Understanding these interactions is essential for comprehending cellular processes.
Up until this point, scientists have focused on DNA, and it wasn't until 2008 when they made strides in sequencing RNA. RNA is the transcribed product of DNA and can provide an understanding of the genes that are being expressed. Here is how RNA sequencing works. First, RNA is extracted from a sample and converted into complementary DNA. This is done by an enzyme called reverse transcriptase, which will create the complementary nucleotide using the RNA as a template. A key difference between RNA and DNA is that an RNA will have an uracil in place of a thymine. This conversion step is necessary because DNA is more stable and easier to work with in sequencing. And now we can use short-read sequencing technology to read the fragments and identify the RNA. Through RNA sequencing, we gain a better understanding of what genes are activated and expressed. Have you ever wondered why different tissues in our body can do different things, even if they all have the same DNA? That's because our DNA is expressed differently in each tissue, and RNA let us capture this difference in expression. During 2009, different methods of long-read sequencing were introduced by different companies in order to sequence longer DNA or RNA fragments. one very well-known method was introduced by Pacific Biosciences. This company created the single-molecule real-time sequencing method. Here is how it works. It starts with fragmenting the DNA up to 2,500 nucleotide pairs or more. That's approximately 8 times more than short-read sequencing. Next, specific adapters are used to bind the two edges of the double-stranded DNA, creating a circular shape of DNA. Then, a primer specific to the adapters and DNA polymerase are added to the PacBio machine for sequencing. Inside the machine, the DNA complex will be transcribed with DNA polymerase, and each nucleotide will emit light that will be measured in real time. As a result, the sequencing machine reads each nucleotide, giving us the final DNA sequence. This and other long-read sequencing technologies have transformed our ability to accurately sequence complex genomic regions that would be hard to sequence with short-read sequencing. It is especially useful to offer resolution of repetitive sequences and complicated DNA structural variants, advancing our understanding of the human genome. So far, we have explored some key advancements in sequencing technologies and how they have drastically evolved. Here are 2 quick questions for you to think about. Pause the screen to think about the answers. In RNA sequencing, why is RNA converted into complementary DNA before sequencing? What are the differences between long-read sequencing and short-read sequencing? If you are looking for the answers, this will be included in the description down below. In 2009, scientists advanced sequencing technology to capture the protein-coding regions of DNA, called exons, in a method called whole exome sequencing. Here is how it works. The process starts, like all others, in which DNA is captured from the sample and fragmented. The difference is using microarrays, which is a surface covered with thousands of spots. Each one of these spots are embedded with a specific molecule called a probe. Probes are known sequences that are complementary to the exons and function to latch onto only exons and not other DNA fragments. Now that we have captured only the exons, this can be sequenced using short-read technologies. This sequencing has been very useful for diseases that have a genetic cause, since exons contain approximately 85% of known disease-causing variants. By looking at just the exons, scientists can capture variants that cause most diseases, while also bringing down the cost of sequencing because exons make up only 1 to 3% of our DNA. In the same year, a method called Bisulfite sequencing was developed to detect DNA methylation. This is a frequent modification that occurs after DNA is replicated, involving the addition of a methyl group to a DNA base, most often cytosine. Here is how it works. Like all others, it starts by denaturing and fragmenting DNA. Next, the sample is incubated with a chemical called sodium bisulfite. When unmethylated cytosines are incubated with sodium bisulfite, these are replaced by uracil, while the methylated cytosines stay the same. Then DNA is amplified with PCR, where uracil will be converted to thymine and methylated cytosines will remain cytosines. After high-throughput sequencing, methylated cytosines will be read as cytosine, and unmethylated cytosines will be read as thymine. This method is useful for detecting DNA regions that are silenced through methylation. methylation can repress gene expression by preventing transcription factors and other regulatory proteins from accessing DNA. By identifying these methylated regions, researchers can gain insights into how genes are regulated. Until 2009, sequencing was typically performed on a bulk mixture of tissues or cells. With the advent of single-cell sequencing, scientists can now zoom into one cell to understand its unique genes and gene expression profile. Here is how it works. To do this sequencing, a major step is isolating single cells from a cell population. Among many methods, fluorescence-activated cell sorting labels uses fluorescent antibodies to bind to specific cells. Cells pass through a single file line, and as each cell passes through the laser, the fluorescently tagged cell will then get sorted out from the other cells and can then further be analyzed through a sequencing method. This is exciting because not all cells are the same. For example, your skin cells have a very different job than your liver cells would. These differences in gene expression between cells are not detectable in bulk sequencing. Now that we have explored all the major sequencing technologies, here are two more questions for you to think about. Pause the screen to think about the answers. How does single-cell sequencing differ from traditional bulk sequencing? In bisulfite sequencing, what happens to unmethylated cytosines after treatment with sodium bisulfite? If you're looking for the answers, this will be included in the description down below. So where are we now in 2024? Now that you have a general understanding of the revolutionary technologies from Sanger sequencing in 1977 to single-cell sequencing in 2009, you'll be surprised to know that all these technologies are still used now. Through this, we have been able to uncover information that has taught us so much about our genetic makeup. And the future is even more exciting. Researchers are now exploring ultra-fast portable sequencing technologies. These advancements will unlock even more information that can advance science and help patients. Thank you for joining us on this journey through the past, present, and future of DNA sequencing.